Okay, a little bit of some new scenery today. Chapter 8, HIV 1, 2, and the Big Bang of Emerging Viruses, AIDS and Ebola, Nature, Accident, or Intentional. The morning after the Stryker interview, I handed Jackie the tape of our conversation. Here, listen to this, I said. I then walked off to my office to do some more reading. A few hours later, Jackie, who had slain much of her fear following the word of Stryker's whereabouts, walked in holding the cassette. This is completely unbelievable, she protested. The part about the HELA cells, aerosolized in labs ending up all over the world, is absolutely horrifying. The whole thing is horrifying, if you ask me, I responded. I have a few problems with what he alleged, particularly the Russian theory, but most of what he said seems plausible. I'm going to check on his references tomorrow. You're going to Harvard? Yeah, I want to read some of the articles he mentioned and see if they have conspiracy of cells. Early the next morning, I made my now routine excursion to Count Way to investigate Stryker's allegations. Unfortunately for humanity, I found scientific evidence to support the vast majority of his claims. His bovine leukemia virus and bovine Visna virus theory of AIDS virus engineering was indeed sound. The methods Stryker cited as those used by cancer researchers to establish new strains of deadly viruses were generally accepted and widely practiced. In addition, evolutionary trees had been developed showing the close relationship between human T lymphocyte virus 1 and 2, the bovine or human T leukemia virus, I believe. Yeah. Well, let's make sure. HTL. Human T lymphocyte leukemia virus. Okay. I was partly right. Okay, now I lost my... Okay. His bovine leukemia virus... His bovine leukemia lymphocyte virus and bovine Visna virus theory of AIDS virus engineering was indeed sound. The methods Stryker cited as those used by cancer researchers to establish new strains of deadly viruses were generally accepted and widely practiced. In addition, evolutionary trees had been developed showing the close relationship between human T lymphocyte leukemia virus 1 and 2 and bovine leukemia virus, as well as between HIV and Visna. See figure 8.1. You guys want to pause it on this? One conflicting report, however, emerged from the National Cancer Institute. The report alleged the risk of BLV or BVV transmission to humans was nil. In a Cold Springs Harbor Laboratory publication, Dr. S.G. Devar wrote, "There is evidence that BLV can be transmitted to sheep through unpasteurized milk. In one instance, leukemia has been reported to have developed in a chimpanzee fed milk from a leukemic cow. However, sera from patients with various forms of cancer, as well as from individuals in contact with cattle, lacked detectable levels of antibodies of BLV proteins. These observations confirm those of others and suggest that BLV is not likely to pose a serious natural hazard to humans." My internal dialogue question again about what about unnatural hazards like intentional mutations and tainted vaccine injections? If chimpanzees were susceptible to cow milk transmitted leukemia, then humans might be also, particularly if the inoculated BLV or BVV was experimentally cultured. in humans white blood cells, as researchers had routinely done. Additionally disturbing was an article I found about human breast milk in Nature New Biology, a journal in which Gallo published much of his work. This report by other members of the National Cancer Institute staff found type C particles, simian sarcoma virus type structures, in mother's milk that resembled known oncogenic RNA viruses isolated from monkey breast tumors. As in Gallo's National Academy of Science report, in which he manipulated simian sarcoma virus RNA to synthesize a DNA probe for human cancer research, the particles found by this group of researchers had an identical density of 1.16 through 1.19 grams per milliliters to the -1 power. What left me cold was the fact that Gallo and others at the NCI were manipulating simian monkey viruses to create human killers, screening American women for these agents, and then oddly enough locating these same particles in their lactates. I couldn't help wondering, might at least some of the world's childhood leukemia cases be linked to nursing infants exposed to National Cancer Institute's simian monkey cancer viruses? Could such experiments have gone awry, exposing American women to breast cancer-causing viruses too? I made note of the fact that the authors acknowledged their National Cancer Institute coordinators, Doctors Brian Maneker and Clausen, for their efforts. Schultz versus Strecker on Montagnier and Gallo. Strecker indicated the Wistar Institute was alleged to have spread AIDS through tainted polio vaccines. In researching background on Wistar, I learned that Hillary Koprowski served as a chief virologist at the facility during the late 1960s and early 1970s. I found it interesting to read that Koprowski also served as co-chair for the Second International Symposium on Tumor Viruses, held at Royaumont, France, on June 3rd through 5th, 1969, during Koprowski's session titled Persistence and Transcription of Viral Genome in Virus Transformed Cells Mechanisms of Induction. He presented the presence of simian virus 40 genome in transformed cells and mechanisms of rescue operation. Luc Montagnier, then with the Institut du Radim, Orsay, France, attended. Montagnier also presented a paper at this symposium, Alterations de la surface des cellules BHK21, and rapport I'm not reading any more French, in which he discussed the nature of changes occurring at cell surfaces following exposure to cancer-causing viruses. In addition, Montagnier was apparently present at the general session in which Doctors Hubner, Toder, and Sarma from the NCI presented switched-off vertically transmitted C-type RNA tumor viruses as determinants of spontaneous and induced cancer, a new hypothesis of viral carcinogenesis. During their presentation, this National Cancer Institute affiliated group acknowledged, as Gallo did shortly after that, their search for cancer-causing viruses was only marginally fruitful. They said, only Burkitt's African lymphoma survives as a cancerous entity, the natural occurrence of which could be regarded as consistent with a foreign virus such as herpes-like Epstein-Barr virus, EBV. Although EBV infections have also been associated with nasopharyngeal cancers, rectal genetic factors, and perhaps religious practices appear to be important contributing factors. Despite extensive Seroepidemiologic blood surveys and experimental tumor induction studies of the oncogenic DNA viruses. None have been established as a significant cause of spontaneous cancers in their natural hosts. These various convincing arguments against DNA and other horizontally species-to-species spread virus as possible significant causes of natural cancer led us to ask this very pertinent question. Do any of the known oncogenic viruses have properties and or behavior patterns consistent with the well-known naturally occurring cancers? We conclude that the only candidates were the RNA tumor viruses of the C and B types, the prototypes of which are the sarcoma and leukemia viruses. Except for the B-type mammary tumor virus, the C-type virus, we believe, represents the only well-established oncogenic group of viruses that can be considered seriously in the etiology of natural occurring cancer, meaning Schultz was mistaken and Strecker was undoubtedly correct. Montagnier apparently knew as much as Gallo not only about slow C-type viruses and monkey viruses, including simian virus 40, but also about the suspected relationship between Epstein-Barr virus and Burkitt's African lymphoma, a white blood cell cancer affecting genetically and behaviorally predisposed black people. Moreover, it was obvious and highly suspicious that Montagnier, Koporowski, as well as Gallo knew that the only candidates for cancer-causing viruses potentially useful as human bioweapons were the RNA tumor viruses, the prototypes that caused the rare AIDS-related cancers, sarcoma, lymphoma, and leukemia. More troubling, these researchers acknowledged cancer studies conducted in New York City that provided evidence of the probable association of virtually all human cancer occurring in different age groups and in different ethnic groups in each of many different years. As Jackie had noted the similarity of the NCI's blood studies to those conducted by the Nazis, here was evidence that New York researchers confirmed as early as 1960 exactly which ethnic group would be most susceptible to lymphoma, leukemia, and sarcoma virus attacks. The IARC and African AIDS. Strecker's theory that IARC brought AIDS to Africa is based on the knowledge that the organization, along with the Cancer Division of the World Health Organization, worked with the International Union Against Cancer, IUAC, a non-governmental voluntary agency devoted solely to promoting the campaign against cancer in its research, therapeutic, and preventative aspects. I'm going to look up what IRAC means. Dang. IRAC. International Agency for Research on Cancer. International Association for Research on Cancer and African AIDS. Okay. The National Cancer Institute reported that the International Association for Research on Cancer was established in 1965 under the auspices of the World Health Organization, initially sponsored by 5 most active NATO countries: the Federal Republic of Germany, France, Italy, the United Kingdom, and the United States, and based on Lyon, France. The IARC, the International Association for Research on Cancer, began work with a budget of $750,000 a year. The budget for 1973 was $3.5 million, as 10 other countries joined the organization, including the Soviet Union. Indeed, when attention began to focus on the apparent link between virus infection and cancers, the International Association for Research on Cancer focused its research efforts principally on Africa. The reasons for this are also suspicious. Despite the existence of cancers in most parts of the world, the International Association for Research on Cancer targeted esophageal cancer and liver cancer, both in black African populations. The National Cancer Institute reported the incidence of cancer of the esophageal shows very wide geographic variations. It has also increased significantly in recent years in certain population groups, notably the black community in the United States, for as yet undermined reasons. In an effort to determine the reasons, an agency is investigating a hotspot for ruminal cancer in cattle, the equivalent of esophageal cancer in man, in Kenya, which affects 10% of all beasts in a very limited area, which may provide clues to the possible etiology of esophageal cancer in those human situations where alcohol is not important. Liver cancer was second on the list of the International Association for Research on Cancer's priorities to determine the relationship between aflatoxin, a toxic chemical produced by the fungus Aspergillus flavus, and liver cancer. The IARC studied individuals in Kenya and elsewhere in Africa. The problem has been further compounded by recent work on hepatitis virus and liver cancer. Serologic studies show that the hepatitis B antigen, HBAG, is associated with 50% of liver cancers in Africa and Asia, two areas of the world wherein AIDS prevalence rates are highest, being found in about 6% of controls. Much lower frequencies were obtained in North America and in Europe, while much remains to be discovered about I just read that. What does HBAG mean? Hepatitis antibodies? Hepatitis B antigen, okay. Much lower frequencies were obtained in North America and in Europe, while much remains to be discovered about hepatitis B antigens. There now exists two putative etiological agents. One chemical and the other viral. Intensive study is needed to show whether they are both in fact responsible for liver cancer, and if so, whether they operate independently or together. A survey of 500,000 individuals has been carried out over 2 years in West Africa to determine the natural history of HBAG. I don't know why I can't remember HBAG. Human hepatitis B antigen, okay. For over 2 years in West Africa to determine the natural history of the hepatitis B antigen. Besides the International Association for Research on Cancer's interest in hepatitis B, even more important perhaps are the sero-epidemiologic studies being carried out with support of the U.S. National Cancer Institute, Bethesda, Maryland, and East African Virus Research Institute. In Entebbe, Uganda, the agency has been studying nasopharyngeal carcinoma in Asia, a Burkitt's lymphoma in Africa neoplasms associated serologically with a herpes virus, a C-type RNA retrovirus. The mass survey has been organized, which will follow a population of approximately 35,000 children in Africa over a sufficient period of time to see whether the development of Burkitt's lymphoma in that population is in fact linked with infection by the virus. The logistic problems are considerable, but no other possible approach has been suggested to date. A map depicting the nature of the International Association for Research on Cancer sponsored programs in Africa and elsewhere was also published in their report. If you guys like to pause it, make sure you get the legend in there. Reorganizing the hepatitis B virus-related cancer research was a concern for the International Association of Research on Cancer as well as Merck Incorporated. I searched to understand why. The answer was apparent in chapter of Prospects for Vaccines Against Cancer, written in the early 1970s by Maurice R. Hillman, chief of the Division of Virus and Cell Biology Research at Merck Institute for Therapeutic Research. Gallo's counterpart at the Merck Labs in West Point, Pennsylvania, Hillman explained the company's interest in viral vaccines this way. It is well established that a great many factors have something to do with cancer: ionizing radiation, environmental carcinogens, age of host, hormones, genetic factors, and the like. It is also well established that a great variety of tumors in animals are caused by viruses, and one needs only to point to the role of viruses in the malignant neoplasia of rodents, cats, frogs, and bovines with ancillary information obtained in studies of cancer in monkeys and man. This leads to the concept that one indispensable element in cancer may be a virus or its genetic material, and all other factors may only be secondary. In taking this position, we are reinforced by the fact that cancer cells seem to have a new genetic input that allows them to make new and unique antigens that are present in the cells and on their surfaces. Carcinogen chemicals and physical agents such as ionizing radiation do not provide such input. They only rearrange the output. If it is true that virus infection is indispensable in cancer, then the prevention of viral infection or the negation of the viral effect might permit the breaking of an essential link in the neoplastic chain, and so make the prevention of cancer possible. This has provided a motivation for seeking the virus of viruses that cause cancer in man. Current research in viral etiology of human cancer has been focused on the C and B type RNA viruses that are being linked with leukemia, sarcoma, and breast cancer, mainly by the demonstration of viruses or virus-like particles in human neoplastic tissues and secretions. To date, however, no reliably propagatable RNA virus has been recovered from human neoplasia that can be considered to be a serious candidate for an etiologic role in man. This in spite of the intense competitive effort given to it and in spite of the private and public proclamations to the contrary. The DNA viruses have fared somewhat better, and the herpes viruses can be seriously considered as a candidate for several neoplasias in man, including Burkitt's lymphoma and nasopharyngeal carcinoma, and possibly also cervical and prostatic neoplasia and Hodgkin's disease. As with RNA viruses, however, proof of etiological role remains elusive and is frustrated by the inability to carry out direct etiologic studies in the human species. Regarding the use of feline leukemia sarcoma model for vaccine production and cancer research, Hillman noted, in judging the probability for developing vaccines against cancer, it is necessary to consider the means for the spread of the virus, whether it be horizontal as in infectious disease like animal-to-animal cross-species transmission, or vertical as in transmission from mother to offspring, either through transplacental infection or through integration in the germ plasm genes. The feline leukemia sarcoma complex presents an excellent model in comparative oncology for testing vertical viruses, horizontal spread of cancer, and for evaluating the effectiveness of vaccines. It is the prime model for RNA oncogenic viruses that is being pursued in our laboratories. The cat might be considered an especially meaningful model for such studies, since the cat, like man, is outbred and it is subject to many, if not most, of the same environmental carcinogens to which man is subject, and which might play a role in the induction of cancer. The rates for spontaneous leukemia and sarcoma in cats are low and roughly in the same range as man. Considering the difference in lifespan, most important, while cats do harbor covert indigenous leukemia viruses, horizontal transmission of the oncogenic virus does occur, whereas the role of the vertically transmitted cancer is in serious doubt. Significantly, adult immunogen. I can't believe I struggle saying this word. Adult immunologic. Okay. In serious doubt, significantly adult immunologically competent cats can be infected artificially or naturally with the virus, and this can result in the induction of leukemia. With further study, it may be hoped that this model will present the means whereby live, killed, and subunit vaccines can be evaluated for prophylactic and therapeutic efficacy, and whereby information can be obtained that can be applied eventually to the development of vaccines for use in man, emphasis added. Finally, Hillman acknowledged the use of simian viruses, two of which were herpes viruses. Hold on. Cow! Come here. Cowgirl! Gotta make sure the dogs stay by. Come on, cow. Come here. Come on. Come on. Hi, cow. Stay right here. Huh? Stay right here. Finally. Oh, hi, Vaughn. Stay there, Vaughn. Sorry. Finally, Hillman acknowledged the use of simian cancer viruses, two of which were herpes viruses. Herpes samiri recovered from kidneys of squirrel monkeys, Samiri sicariusis, and herpes atilius isolated from kidney culture of a black spider monkey. I'm not even going to attempt to say that word. I guess it's the Latin word for spider monkey. I ain't trying. Known to cause lymphoma or rectilium cell tumor and leukemia. When injected into other monkey species, these viruses, Hillman wrote, present excellent models for studies for immunologic interruption of cancer in primate species using live, killed, or subunit viral vaccines. They are of special importance because of their possible use in elucidating the interactions among Epstein-Barr virus, Burkitt's lymphoma, and nasopharyngeal carcinoma in man. Now I better understood what Nixon's war on cancer and the cancer virus hypothesis was really about, the search for the virus and vaccine. I also realized why the generals on the battlefield, Gallo at the National Cancer Institute, Hillman at Merck, and Don Francis at the CDC, were fooling around with leukemic cat and simian monkeys. These models, they claimed, might provide the most powerful weapons against human immune system destroying germs. Tribute to Don Francis. Attempting to investigate Strecker's claims that Dr. Gallo Francis and Montagnier were longtime workmates at the National Cancer Institute, I sought Francis's biography in American Men and Women of Science. In a nutshell, here's what I learned. Donald Pinkston Francis was born in Los Angeles, California on October 24, 1942. He married in 1976 and had two children. Educated at Northwestern University, he received his medical degree in 1968 and then an honorary Doctor of Science degree from Harvard in 1979. His specialty was noted as medical epidemiology and virology. From 1978 to the time of this writing, he served as chief of the epidemiology branch at the Hepatitis Lab Division of the CDC. Francis's professional experience included service as an international fellow in the Children's Bureau of the U.S. Department of Health, Education and Welfare in Punjab, India. In 1968, internship and residency in pediatrics at the Los Angeles County University of Southern California Medical Center, 1969 through 1971. United States Assistant for International Development Service as a state epidemiologist in infectious disease in River State, Nigeria. In 1971, American Epidemiologic Intelligence Service Officer for the CDC, 1971 through 1973, and Medical Officer for the World Health Organization smallpox vaccine program in the Sudan, which abuts the northern border of Zaire and Uganda, the hot zone wherein the AIDS, Ebola, Zaire, and Ebola Sudan viruses allegedly jumped species 3 years after Francis's tour of the Sudan ended. He then went to India from 1973 to 1975 and finally received an infectious disease fellowship at the Channing Lab at Harvard Medical School from 1975 to 1977. He currently holds a position at my alma mater, Harvard School of Public Health, HSPH, Harvard School of Public Health, and he began as a fellow in microbiology in 1976. His mailing address was listed as the Division of Oral Disease, CDC, Atlanta, Georgia 30333. I found nothing to indicate Francis and Gallo shared common workplaces in the early 1970s. Although Francis received several grants from the NCI during that time, he also shared with Gallo several acquaintances, including Max Essex at the Harvard School of Public Health, Department of Microbiology, with whom he co-authored many feline leukemia virus reports. Consistent with Streckert's allegation of collusion, however, it's hard to imagine this most esteemed group of American type C RNA retrovirologists were unaware of each other's studies and findings during this important time in cancer virus research. Given his unique early 1970s cat leukemia virus research, hepatitis B virus studies, medical intelligence background, and third world service record, the fact that Francis noted the laundry list of cat leukemia virus sarcoma virus symptoms among gay men and intravenous drug users in the early days of the AIDS epidemic made me question his knowledge and service record even more. Combine these two diseases, Francis said to a gathering of colleagues, feline leukemia and hepatitis, and you have the immune deficiency. Surely Francis knew what Hillman had clearly explained. He would have known that Gallo had taken monkey viruses, extracted their humanly benign DNA, infused their empty viral shells with cat leukemia sarcoma RNA, and then cultured the genetically engineered mutants in human white blood cells to allow them to cross the species barrier. The CDC's hepatitis chief might have an inkling that this new retrovirus he suspected of causing GRID, gay-related immune deficiency, I believe that's what it was, might have come from Gallo's or Hillman's labs or another National Cancer Institute research group that manipulated viruses in this way. Particularly, he, Gallo, and Essex presented their cat retrovirus research at the same session of a 1980 Cold Springs Harbor conference, in which cat leukemia and sarcoma virus mutations, cloning the cross-species transmissions, were discussed. If Schultz were alive, he might now realize an alternate reason why Francis was particularly incredulous that the National Cancer Institute had successfully stalled the AIDS research effort. Could their stalling have provided time to organize and administer an effective disinformation campaign? Surely Francis knew more than what he was saying. Francis in Ebola territory. I found it interesting that Francis directed the emergency response team sent by the WHO to control the outbreak of the dreaded Ebola virus. Schultz chronicled this activity as well. The horrible fever had swept seemingly from nowhere into the border region between Zaire and Sudan. On the fetid banks of the Ebola River, the disease was a blood-borne virus, wickedly spreading both through sexual intercourse because infected lymphocytes were in victim semen and through the sharing of needles in local bush hospitals. The absence of modern precautions to protect doctors also spread the blood-borne disease among medical personnel through routes unimaginable in more civilized countries. During this 1976 outbreak, local Danish doctors in the remote hospitals in Zaire, people like I.B. Bierbjerg, the Gerthrask, were impressed with the vigor with which the team from the World Health Organization had moved to stamp out this deadly disease that became known as Ebola fever. When it became obvious that the disease was spreading through autopsies and ritual contact with corpses during the funerary processes, Dr. Don Francis, on loan to the World Health Organization from the CDC, had simply banned local rituals and unceremoniously burned the corpses. Infected survivors were removed from the community and quarantined until it was clear that they could no longer spread the fever. Within weeks, the disease disappeared as mysteriously as it had come. 6 years later, on January 6, 1982, Francis could not escape the memories of the horrible Ebola fever outbreak, wrote Schultz. His recollections became more acute when he received a phone call from Dr. Guy in the Paris. His recollections became more acute when he received a phone call from Dr. Guy D.M.D. in Paris, another veteran of African epidemics. That must be a strange name. Dr. D. I guess it is his name. Dr. D. had studied the latest data from Africa. Common cases of benign Kaposi sarcoma were not in question. Typically, Kaposi sarcoma in African males responded to treatment. Francis, however, had already heard of the new, more virulent KS that had been reported first in Uganda in 1972, but there was more. Ditha said, in the Western Nile district of Uganda, young men living together were getting not only the typical easygoing Kaposi sarcoma, but the nasty kind, like the tearing through the bodies of American homosexuals. These Africans also suffered from the lymphoadenopathy that marked the early stages of the American disease. Said there had to be some connection, emphasis added. Of course, Francis thought, a new virus from Africa. It was where Bob Gallo and the National Cancer Institute figured his new retrovirus for human T-cell leukemia came from, too. After all, the human T lymphocyte lymphoma virus only struck in the portions of Japan settled by the Portuguese traders who apparently had brought the microbe with them from Africa some 500 years ago. The African links reinforced Francis's hypothesis about a transmittable agent, but the National Cancer Institute didn't seem terribly interested in the disease. If I struggle with these sometimes, there's a lot of abbreviations in here, and I'm doing this all from memory, and sometimes I have to refer back to the definitions page, so bear with me. Come on, Schultz, I respectfully implored the diseased journalist. You were a lot smarter than that. Why would it have taken 500 years for an African virus to express itself in Japan while everywhere else in the world it reared its ugly head in less than a decade, 90% of the time? Had Gallo figured his new retrovirus came from Africa because that's where the experiments were taking place in the race to create special cancer viruses and vaccines? A race in which he, Francis, and Hillman were majorly contestants, a race to create dozens of leukemia sarcoma RNA retroviruses for vaccine tests in black Africans in the Nile district of northwest Uganda and American homosexuals in New York City. Finally, I found it interesting that following a successful stint in Nigeria as a United States Aid for International Development, infectious disease researcher, and a couple of years in Sudan on the behalf of the CDC and WHO as a smallpox vaccination program officer, Francis was assigned a principal role in the gay male hepatitis B vaccine study funded by the CDC and Merck. Sharp and Dome. Notes on Duesberg. As I was searching the literature, another prominent name in AIDS research circles kept appearing. Peter Duesberg. As detailed in Deadly Innocence and elsewhere, Duesberg is regarded as an esteemed American scientist, a member of the National Academy of Science, and is credited for having initially mapped the genetic structure of retroviruses. Unlike most AIDS scientists, however, Duesberg has since the mid-1980s argued that AIDS is completely atypical of an infectious disease and not likely caused by retroviruses, Gallo and Montagnier identified. How is this possible, I questioned during the Deadly Innocence investigation. To be an infectious disease, traditionally, scientists had to prove a cause-effect relationship between the suspected germ and the symptoms it is alleged to cause when the germ spreads to other hosts. As Strecker alluded, these standards, known to Koch's postulates, are based on 3 tests. First, the suspected germ must be found in everyone with the disease. Not all people with AIDS test positive for HIV infection. These patients would now likely be diagnosed as having idiopathic lymphocytopenia, ILC. It's amazing I can say these huge words and mess up simple words. A disease in which white blood cells spontaneously disappear for no apparent reason. Second, the germ should not be present in individuals without illness. Yet, of the esteemed 1 million Americans who are believed to be HIV carriers, 3/4 have not developed AIDS. Furthermore, a recent CBS News report, scientists noted that 8% of those who carry HIV antibodies have remained symptomless for more than a decade, suggesting that they may be carrying a weaker strain or doing something special to prolong their health. Positive thinking, exercise, and good nutrition may be part of that special something. Third, the researchers should be able to reproduce the illness in laboratory animals using isolated germ cultures. Duesberg argued that, with AIDS, this has proved difficult. In the August 5, 1992 issue in These Times, health writer Benjamin Goldman and AIDS journalist Michael Chappell reported that defenders of the HIV hypothesis simply rejected Koch's postulates as being outdated. They stated that Harold Jaffe, a senior AIDS HIV investigator at the CDC, and Robin Weiss, a British AIDS researcher, argued, "What seems bizarre is that anyone should demand strict adherence to these unreconstructed postulates 100 years after their proposition." Reporters Goldman and Chappell, however, noted the advice of Nobel laureate Walter Gilbert of Harvard University. Someone scientifically trained will not make that statement. Koch's postulates are an attempt at rigorous proof. If you cannot fulfill Koch's postulates, you got a problem. You can deal with that either with hard thinking or with soft thinking. If you can block the virus and thus block the disease, that would constitute hard evidence that you were right. But in the absence of a successful treatment, Gilbert characterized the attempt to dismiss Koch's postulates and fall back on incomplete epidemiological statistics as soft-minded. I questioned, might Koch's postulates, the scientific test to prove etiologically of naturally occurring infectious diseases be outdated and dismissed only because they don't hold for unnatural man-made infectious diseases? In any case, Duesberg has argued for more than a decade that you can find HIV in any burned out, drugged out, and sexed out individual. He has consistently blamed the victims of AIDS for living unhealthy lives. I'm sure that's a major proponent. His arguments are difficult to dispute since physicians and scientists have known for years that strong host resistance is a key factor in determining whether or not an infectious germ can take root, grow, and ultimately overwhelm the immune system. Moreover, he has claimed that HIV is very difficult to spread, which it is. With rare exception, it generally requires larger doses of viruses in more healthy individuals to claim lives. So nothing Duesberg said is new. In fact, Duesberg's position is exactly that held by the prominent NCI and NIH researchers, National Cancer Institute and National Institute of Health researchers from the beginning of the epidemic. As Schultz wrote, it is incredulous that the National Cancer Institute in 1982 was still fiddling around with half-baked theories that gay-related immune deficiency was caused by poppers or sperm. But those were the pre-sensations that NIH researchers made at the conferences. None of them was talking about what Francis thought was the most obvious cause, a new viral agent. The fact is that Duesberg, like Gallo, Montagnier, Francis, Hillman, and a few others, had been part of a core group of investigators funded by National Cancer Institute to study special viruses and their links to cancer. In 1971, for instance, Duesberg became a project director for the University of California, contract NIH 71-2173. See, here. You want to pause it and read it yourself. Be my guest. To study the mechanisms regulating the mutant RNA tumor virus reproduction and sarcoma development, the RNA-dependent DNA polymerase-related effects Gallo and his team studied. The work followed sarcoma and leukemia virus studies on monkeys and cows, also conducted at the University of California with NCI funding. The same year, during a meeting in Paris by Gallo and Montagnier, Duesberg presented evidence that RNA leukemia and sarcoma viruses can be manipulated and observed to produce the provirus DNA needed to successfully reproduce in nature and cell cultures. Earlier, Duesberg evaluated the effects of chemotherapy using the antibiotic Actinomycin D on mouse leukemia virus, MLV. He later examined leukemia and sarcoma viruses to show how mutants of varying oncogenesis are formed. Most pertinent to Duesberg's main arguments, human T lymphocyte lymphoma virus 3. Was it lymphoma or leukemia? I'm just trying to have this solidified in my mind. If it's not solidified in my mind, how's it going to be solidified in yours? Maybe you got better memories. Human T lymphocyte leukemia virus. Most pertinent to Duesberg's main argument, human T lymphocyte leukemia virus 3 does not cause the immune deficiency and cancer seen in AIDS. Duesberg and Gallo in 1973 debated essentially the same question: can special type cRNA tumor viruses, such as those captured from simian monkeys and modified for human experiments, cause cancers? Duesberg answered affirmatively. That is absolutely right. Here's figure 8.3. NIH report shows Peter Duesberg at the University of California in 1971 directed studies which paralleled Dr. Gallo's at the National Cancer Institute. Duesberg intended to explain the mechanism by which RNA tumor viruses bring about malignant changes in humans. Mutant sarcoma viruses were used for this purpose following studies inoculating cows and monkeys with simian sarcoma and leukemia viruses by Leo Bustad and Robert Hubner at UC and the NCI. This evidence, along with a conference discussion, raised serious questions regarding Dr. Duesberg's more recent publications asserting HIV cannot cause AIDS. Source: NCI staff, Special Virus Cancer Program, Progress Report 8 and 9, Office of the Associate Scientific Director of Viral Oncology, OASDVO, J.B. Maloney, Ed. Washington, D.C., U.S. Government Printing Office, 1971, page 233. The discussion occurred following Gallo's presentation of the origin of human acute myeloblastic leukemia, the virus hotspot hypothesis. Gallo was joined by Dr. Wu, who presented two related papers as an emissary of Littleton Bionetics. During the session's open discussion, Gallo questioned whether the genetic hotspot that caused cells to become cancerous was responding to an external or internal virus-like particle. Gallo: Are there viral type C RNA tumor virus genes in some normal cells? Everyone by now must believe that there are some viral genes in at least some normal cells. I would like to know where they came from, or which came first. Are these viral genes in fact really cell genes which the virus utilizes? Duesberg should speculate on this. Duesberg: That's too much for me. That's like all theories on the origin of life. Where do whales come from? Where does God come from? Where does a clean chicken come from? But I would like to return a question to you, maybe somewhat related to that. I think we can divide those viruses which cause cancer and those which are subvirus-like things which may be a consequence of cancer. I think that those which are causing cancer may be like the men and the other more or less like the boys. So I think that shouldn't be confused too much. I think these subviral particles or endogenous viruses or incomplete endogenous viruses or enzymes might in fact well be a consequence of cancer rather than its cause. But I think there is little doubt that Rous SV, mouse sarcoma virus, or AMV, bird virus, can be the cause of cancer. Gallo: I kind of agree with that. At least they cause chicken cancer. I think the information for carcinogenesis may be packaged into only very special type C RNA tumor viruses. But I wouldn't even make those viruses that you call boys any less important, because boys can become men. Moreover, we have now demonstrated that the reverse transcriptase in human leukemic cells and the viral-related nucleic acid is related not to endogenous non-oncogenic type C viruses, but specifically to type C viruses, which in fact are oncogenic, such as the woolly monkey simian sarcoma virus. Duesberg: That's absolutely right, emphasis added. In essence, Gallo stated that Duesberg acknowledged that only very special retroviruses could be expected to produce AIDS-like symptoms, and this had already been proven in monkeys and other animals. In contrast, since the publication of his objections to HIV equals AIDS paradigm in 1988, Duesberg has been diverting public attention from the fact by arguing that HIV has nothing to do with AIDS. AIDS-like viruses do cause human immune system disorders and cancer, he admitted, at least in chickens and monkeys before they were clearly modified to infect human cells. In light of Duesberg's documented ties to the National Association of Science and National Cancer Institute at the exact same time, the NAS formed the D.O.D., that AIDS-like viruses could be developed in 5 years for $10 million. I wondered whether the professor intended to provide a disinformation smokescreen for these organizations. Earliest AIDS cases. One important consideration remained before the basic premises of man-made HIV development could be reconciled. The literature held several accounts of alleged AIDS cases and HIV discoveries, predating the work of Gallo, Hillman, Duesberg, that is, prior to the 1960s or 1970s. A literature search conducted to investigate such claims revealed several interesting discoveries. I read a series of reports published in The Lancet regarding the earliest alleged AIDS cases. A 25-year-old former naval seaman who died of cytomegalovirus and pneumocystis infections in 1959. The claim was made by University of Manchester researcher Gerald Corbett and co-workers, having meticulously extracted the seamen's tissues from paraffin blocks initially developed to make autopsy slides. DNA amplification methods were used to search for very small quantities of HIV proviral DNA in targeted cells. The group found the suspected particles in several of the recaptured tissues, and the news media broadly heralded the event. Several years later, Dr. David Ho and Tofu Zhu of the Aaron Diamond Research Center in New York re-examined Corbett's tissue in an effort to investigate the theory that HIV evolved somehow from tainted polio vaccines during the 1950s. Ho and Zhu determined that the DNA sequences found by Corbett and his colleagues were essentially identical to a strain of HIV circulating in the United States during the late 1980s. This raised the specter of specimen contamination. This contamination, the evidence showed, was more likely to be caused by another clinical specimen than by improper DNA probing techniques. And when additional methods were used to learn more, the New York researchers concluded that the tissues examined were derived from at least 2 individuals. Thus, the Corbett group's finding was invalidated. Corbett, allegedly mystified by what Steve Connor of London's daily The Independent called a calculated hoax or mammoth error, later requested the material be re-examined by a third party. It's a big chapter today. Okay. HIV in Zaire, 1959. Another study supported by grant obtained by Robert Gallo and Don Francis's colleague Max Essex and his Harvard associate P. Kenke alleged to have found one HTLV3 HIV positive plasma specimen among 1213 from Central Africa dating back to 1959. Much ado accompanied this find, especially since it was said to have come from Zaire. Essex's group also reported that the presence of HIV in the sample had been confirmed by 3 other laboratories using different techniques. According to scientific consensus, however, an independent confirmatory test was never done. 3 other laboratories that Essex alleged checked his group's work may not have been impartial. These included Gallo's lab in Bethesda, Dr. C. Schabel's lab at the CDC, and Abbott Laboratories. Abbott Labs is best known for having licensed and produced the ELISA screening test for HIV. This test had also been used by Saxinger, Gallo, and others who claimed 60% of Ugandan children were infected with HIV by the early 1970s. This report was later debunked by several research groups that noted the esteemed NCI researchers had failed to use HIV-specific testing methods or materials. Abbott also licensed and marked the hepatitis core antigen test purchased by New York City Blood Center officials following years of delay before the ELISA test was available to help identify blood units suspected of HIV infection. The company had also supplied expertise and the radioactive experimental reagents Zemusis required for his New York homosexual hepatitis B vaccine trial. Furthermore, Abbott Labs ended up commercially marketing MSD hepatitis B vaccine. Moreover, the hepatitis B vaccine, suspected of having transmitted HIV to American homosexuals, was research done by Abbott L.J. Overby, who was intimately connected with the NYUMC hepatitis B chief Saul Krugman. Together they evaluated hepatitis B susceptibility and vaccination methods in the New York subjects during the mid-1970s. As it turned out, additionally confirmatory studies could not be carried out by independent investigators, as the specimens containing the Leopoldville strain HIV had been lost by the Essex team. AIDS case in 1968. In 1984, researchers published evidence that Robert R., a 15-year-old black male, heterosexual, born and reared in St. Louis, had died of AIDS-like illnesses. Later in 1987, scientists provided evidence that the boy's blood contained antibodies to HIV. If a virus related to HIV, they concluded, has been present in the United States, Africa, or elsewhere for several decades, its failure to spread in an epidemic fashion earlier may reflect a recent genetic change in the virus and/or sociocultural factors involving sexual practices or numbers of sexual partners. By 1990, advanced gene analysis techniques allowed scientists to re-examine this case, at which time they determined that Robert R. had not died of AIDS. His blood never contained HIV. This evidence and more led the consensus of international AIDS researchers to conclude that some radical event between 1970 and 1975 had changed the HIV-1 progenitor from a virtually harmless germ, essentially non-infectious to humans, to a silent killer. Dr. Gerald Myers, chief of the Special HIV Sequence Database AIDS Project of the United States government's Los Alamos National Laboratory, may have articulated this position best when he said, "The preponderance of evidence still argues for an explosive event in the mid-1970s. Furthermore, regarding the origin of AIDS, he insisted HIV-1 was a fairly new virus, surely only a few decades young. Alternatively, Ho and other virologists proposed that HIV-1 may have been ancient, but the mass epidemiological evidence indicates a dramatic change occurrence in the mid-1970s, most likely due to human events more than biological ones in the natural evolution of the virus. What might those obvious human events have been? Obviously, they would have needed to take place simultaneously in North America and Africa. An Hydrogenic event involving vaccines being tested on both continents on monkeys and people offers the most plausible and, in fact, only explanation being advanced." Yeah, take your vaccines, people. Polio vaccine theory of AIDS. Chief among these theories, I learned, is the polio vaccine theory, a fascinating article entitled Simian Retroviruses, Polio Vaccine, and Origins of AIDS by Walter Kyle sparked a flurry of debate in The Lancet. Kyle, a New Hampshire attorney who represented a client who had become paralyzed, apparently from contacting someone who had received the polio vaccine, raised a number of concerns. Chief among them was the fact that live SV40 simian virus that Gallo, Koporowski, and many others had manipulated for cancer research had been known by government officials and vaccine manufacturers to contaminate all Salk inactivated vaccine, as well as Sabin original seed strains. Max Essex and his colleagues, Kyle noted, reported that the African green monkey, the species used in the production of most live polio vaccine in the U.S., was a reservoir of simian immunovirus. Presumably, the regulatory authorities concluded that the presence of any such monkey virus would not affect man because there would be no transfer from the digestive tract to the lymph and blood systems, and because there was no reason to suspect interspecies transfer. At that time, this must have seemed correct. Otherwise, there would surely have been an outbreak in the child recipients of the vaccine. Thus, government and industry standards overlooked SV40 and SIV vaccine contaminants in the range of 100 viruses per dose. Kyle then explained that during the mid-1970s, homosexual men in the U.S. were prescribed SIV-tainted polio vaccines in effort to treat their herpes infections. Indeed, Kyle's theory was so plausible that researchers around the world, including some ordered by the Food and Drug Administration, began testing suspected vaccine lots for type C RNA tumor viruses. Kyle concluded, my hypothesis that the virus particles found in those vaccine lots were HIV or some variant can be tested by analyzing stored samples by the polymerase chain reaction, PCR. Reverse transcriptase analysis of released vaccine have shown up positive for such simian viruses up to 1985, and a critical look should now be taken at all such vaccines. If U.S. government laboratories have already done PCR tests on stored samples of the incriminated lots of polio vaccine which remain, the results should be made public. Several rebuttals to Kyle's controversial report were subsequently published. Some investigators expressed the opinion that the origin of AIDS is unimportant and that research time and money might be better spent finding a cure. This led Raphael Stryker and Blaine Ellswood, two Californian scientists, to respond that Kyle's work, though hardly surprising, was very important for three reasons. The sociological reason is that victims of the disease should not be blamed for starting it. The scientific reason is that new therapies for AIDS could be developed from an understanding of its origin. And the ethical reason is that the sequence of events culminating in AIDS should never be allowed to happen again. Kyle's theory also brought praise and criticism from Thomas F. Schultz of London's Institute of Cancer Research. It was legitimate to ask whether one advanced in medicine, such as the highly successful vaccines against poliomyelitis, might not inadvertently have given rise to another catastrophic AIDS, he wrote. However, there were reasons why Kyle's theory was questionable. Foremost among these was Schultz's concern that African green monkey immunodeficiency viruses, SIV, are not C-type viruses. They differ significantly from HIV, so much so, Schultz argued, that they would not be suspected of causing human AIDS, even though they had contaminated the polio vaccine lots. Schultz and The Lancet omitted, however, was the fact that initial reports of the discovery of HIV and SIV classified them as type C viruses. Kyle relayed during a telephone interview, although now called lentiviruses, they go through a stage of type C morphogenesis during replication and look similar to the type C viruses. Additional support for Kyle's theory came in the Journal of Virology. RNA viruses in general and lentiviruses in particular undergo extensive genetic variations as a result of error-prone replication and recombination, such that they are considered to exist as quasi-species. That is, a population of relatives with similar genes. One researcher noted the exceptional ability of HIV-1 to mutate results in rapid development of quasi-species which evade host defenses and become resistant to various antiviral agents. Furthermore, supporting Kyle and relating to Gallo's early work on the development of AIDS-like viruses, researchers later observed that HIV undergoes type C morphogenesis. Although not classified as type C viruses, lentiviruses follow a similar assembly strategy, by which capsid shell formation and budding of virus from the infected cell occur simultaneously. SV40 shell, likewise, is called a capsid, I recalled, perhaps not coincidentally, then, during the metamorphosis of HIV, researchers found a reproducible peak of viral protein in the fraction corresponding to a density approximation of 1.15 to 1.16 grams per milliliter in gradients of GAG HIV. That is the gene that codes for the inner shell capsid-like structure of the AIDS virus. The number 1.6 jogged my memory. Gallo reported it also, but in 1973, after repeatedly covering the same destiny virus-like particle from human leukemic cells that was capable of producing the principal rapidly growing cancers as seen in AIDS, including sarcomas, leukemias, and carcinomas. The FDA's Bureau of Biologics, as Kyle noted in The Lancet, also found a similar characteristic in the adventitos virus. I'm going to call it the adventitios virus. Found in some live polio vaccine approved by FDA and released in 1977, could the SV40 capsid have become the inner shell of a more complex retrovirus following the recombination with SFV and FELV? Let's look up what that means. FELV, feline cat leukemia virus. SFV. Simian foamy virus. Yes, simian foamy. Okay, so could the SV40 capsid have become the inner shell of a more complex retrovirus following the recombination with simian foamy virus and feline leukemia virus, chicken leukemia, and perhaps other viruses? The scientific evidence was mounting. Furthermore, Schultz explained the differences between HIV-1 and HIV-2 and SIV and made a very important observation. HIV-2 is virtually identical to a monkey virus found only in captive macaques. The closest primate relative of HIV-1 is a lentivirus isolated from chimpanzees, SIV, and the closest relative of HIV-2 found in free-living monkeys is SIV from pseudomangabes. Both HIV-2 and SIV are closely related to SIV from macaques, a virus found only in captive macaques and one which may have been transmitted to this species in captivity. All this means that it is uncertain whether the viral particles observed by electron microscopy and reverse transcriptase assays, as Kyle reported in certain polio vaccine lots, were indeed SIV. They may have represented the endogenous retroviruses, retroviral genomes carried in germline for millions of years and activated and packaged into virion particles under the conditions of tissue culture, emphasis added. In other words, the vaccines may have been tainted with monkey virus genes that for millions of years posed no threat to humans, but had acquired HIV-like capabilities, including the capacity to produce AIDS in humans, while being accidentally or intentionally altered in laboratories during human tissue culture processing. Therefore, Schultz agreed that it was important to take up Kyle's suggestion and examine any remaining vaccine lots by the polymerase chain reaction that might identify HIV or related lentiviruses. Significantly, Kyle observes a more recent Ho et al. I don't know what that et al. Means. Report that HIV-2 primarily appears in the wild in sexually active adult pseudomangabes, not in immature monkeys, a fact which additionally discredits the evolutionary origin of AIDS theory. Finally, Schultz commented that the Ellswood-Strecker theory that certain live poliovirus vaccine lots dispensed in Zaire in 1957 and 1960 and prepared on African green monkey kidney cell cultures could have been inadvertently infected with monkey lentiviruses hypothesized to represent the ancestor of HIV-1. While this theory was compatible with the idea that the AIDS epidemic began in Africa, Schultz reiterated his belief that the AIDS agent could not have been SIV. Suspecting, however, a related hybrid or intermediary might have given rise to HIV-1. During vaccine manufacture, he wrote, "It would be of scientific and practical interest to identify the retroviral particles present in some of the polio vaccine lots. The same might be said for hepatitis B vaccine lots used on New York gay men, I realized." Hold on a second. Callie! There you guys are. Good dogs. Max Essex HIV-2 and the origin of HIV-1. Perhaps not coincidentally, then, the discovery of monkey AIDS virus dates back to the earliest days of recognized human epidemics. According to Laurie Garrett's text, The Coming Plague in 1969, researchers at the California Primate Research Center in Davis witnessed the first outbreak of AIDS-like symptoms among 42 macaques. The monkeys suffered severe T-cell immune system depression, lymphomas, Kaposi's like skin patches, and a host of opportunistic infections. Two similar outbreaks occurred in 1976 and 1978 in the same facility. Next, Garrett chronicled another French-American AIDS fracas, this time between Luc Montagnier and Max Essex, regarding HIV-2, initially dubbed HTLV-4 by Essex's group and LAV-2 by the French. Human T lymphocyte leukemia virus 4 by Essex's group and LAV. What is LAV again? LAV. Lymphedemiopathy associated virus. So lymph node virus. Essex and colleagues alleged discovering the virus among healthy Senegalese female prostitutes. They reported the women's immune response to HTLV-4 and SIV were equally strong. Essex claimed this find was the missing link to HIV-1. It was very similar to the monkey virus and humanly benign. Eventually, Gallo published a detailed genetic analysis of SIV and HTLV4 that indicated these viruses were identical. Presumably, Gallo's group argued Essex's laboratory had been contaminated. Monkey and human tissue were somehow mixed. Yet, how could SIV and HTLV4 and HIV2, identical new retroviruses determined to be laboratory contaminants by Schultz, Gallo, and others, with roots in the monkey virus kingdom, be infecting Senegalese women and not Mackays in the wild? The laboratory was the only common denominator, I realized. For HIV-2 to infect Senegalese women and only humans in the wild, not monkeys, the carriers would have needed to be exposed hydrogenically, that is, by the hand of man. Vaccines were the most plausible way, particularly since SIV, a documented vaccine contaminant, was also present in these women. Later, during the 1996 National AIDS Update Conference held in San Francisco, I had the opportunity to ask Max Essex during his press conference how, other than through vaccines, could HIV-2, a laboratory monkey virus contaminant not found in monkeys in the wild, have gotten into his Senegalese subjects. After beating around the bush for several minutes, unwilling to answer the question directly, he defended, "I can tell you how my monkeys got infected. Researchers had inoculated the monkeys with human tissue during experiments unrelated to HIV prior to them coming to my lab." Oh, we vaccinated them. Oh, we inoculated them. Yes, we'll use that word. Almost done, guys. Though Essex's comment failed to explain just how HIV-1 and HIV-2 got into black Africans and gay Americans in the first place, it did provide an insider's view of human error commonly associated with laboratory contamination, during the threat of viral contaminants being spread from human to monkeys and back again. Indeed, the laboratory was the only common denominator, I realized, and experimental vaccines were the most likely transmitter. The Big Bang Theory. Various researchers determined that HIV-2 preceded HIV-1's evolution, as family trees were constructed for these related viruses based on archaeoepidemiology, or what others call molecular epidemiology. Scientists concluded the two HIVs shared a recent common ancestor, SIV. This is figure 8.4. The evolution of AIDS-like viruses based on gene typing and molecular epidemiology theory as presented by Dr. Gerald Myers, chief of the specialist HIV sequence database AIDS project at Los Alamos National Laboratory. Myers stated the preponderance of evidence still argues for an explosive event in the mid-1970s regarding the origin of AIDS. He insisted HIV-1 evolved fairly recently from SIV. SV40, the monkey virus Gallo and Bionetics researchers genetically altered in a series of steps and then cultured into human white blood cells to alter its outer membrane characteristics, may have been a building block for HIV-2 and HIV-1. Additional evidence suggests that SIV may have been man-made as well. Source Seach Chapter 6 and Myers McInnes K. and Myers L. Phenogenic Moments in the AIDS Epidemic, Chapter 12, in SS Morse ED, Emerging Viruses, Oxford, English, Oxford University Press, 1993. Here's the diagram. Pause it if you like. During the 1990s, it was discovered that HIV-1 and 2 were about 43% homologous or similar to SIV, and HIV-1 were about 43% alike. 3. SIV and HIV-2 and SIV were genetically 72% alike, and 4. SIV and SIV AGM envelopes were 91.4% homologous. I considered this information along with the phylogenetic tree developed by Gerald Myers, see 8.4, I just showed you that, and realized that Myers' mid-1970s Big Bang Theory coincided with one, the completion of efforts on behalf of the D.O.D. to create AIDS-like viruses for biological warfare, and two, the work of Gallo and Littleton Bionetics researchers as diagrammed in Figure 6.5. I wondered whether a similar series of SIV cancer virus vaccine experiments might have resulted in mutations and phylogenetic variations consistent with all the findings above, including those reported by Schultz and Kyle regarding the possible iatrogenic origin of HIV-2 and SIV, HIV-1, and AIDS. Hydrogenic is just the thing of getting a vaccine. Okay, let's look at the Idahogenic theory on the evolution of HIV-1 and HIV-2. A new theory on the origin of AIDS from simian virus progenitors modified through laboratory experiments diagrams shows HIV-1 most likely evolved from the virus Gallo et al. Were genetically altering, then culturing in human white blood cells to produce viral vaccines tested in monkeys and humans during the early 1970s. Other researchers have reported that HIV-2/SIV MAC, found only in macaques, maybe that's what it means, SIV macaques, only found in macaques in captivity and humans in the wild, is the missing link. That is the progenitor of the AIDS virus. HIV-1 then likely resulted from tissue experiments and contaminated vaccine trials. Additional evidence supporting this theory was reported by Narayan in 1995 from the Kansas University Medical Center based on hybrid virus studies by the NIH. I realized that by genetically altering monkey viruses and monkey virus vaccine contaminants, as many researchers, including Gallo and those at Bionetics, had done prior to culturing them in human tissue, they might have created AIDS virus progenitors like SIV and HIV-2, and others to which Schultz and Kyle were referring. Putting all the facts together, I now understood how humanly benign DNA monkey viruses like SV40, SIV, AGM, and other common retrovirus vaccine contaminants like SFV could have, over the period of a few decades, become RNA retroviruses that, through contaminated vaccines, spread to millions of people around the world. SV40 may have been a building block for SIV, which was 72% identical to HIV-2 and 43% like HIV-1 AGM. The highly unstable monkey retrovirus SFV, or other possible contaminants, including the leukemia, sarcoma, and immunodeficiency mutants Gallo and others put together, could have easily recombined with SV40, SIV, AGM, or SFV together. They might have undergone the laboratory transformations and vaccine transmissions resulting in the AIDS pandemic today. In late 1995, Bill Narayan, a Kansas Medical Center virologist, reported a similar transformation in reverse, making SIV that can cause monkey AIDS from HIV-1. He used a hybrid of SIV and HIV-1 that contained the core of the monkey virus and the outer coating of the human virus. This hybrid was supplied by Max Essex's group at Harvard. Narayan's study funded the philanthropic foundation of the Hochst Marianne Rosell Incorporation, formerly known as Marianne Merrell Dow Incorporated, was heralded as important for developing a new vaccine against AIDS. The study was later awarded a $1.6 million grant from the NIH. Indeed, the Hydrogenic origin theory of AIDS, the synthetic development of HIV-1 and HIV-2, and a host of other killer viruses seemed most plausible. And discussion, see figures 8.5 in discussion in Chapter 6. I just showed you 8.5. After digesting all these facts, I concluded that claims of HIV's existence prior to the 1970s had been seriously questioned, and those who made them, particularly Gallo and Essex, appeared to have on several occasions incriminated themselves. Up to my neck in this world of cancer research, scientific misconduct, and counterintelligence, it seemed very hard to distinguish incompetence from duplicity. Whew. Isn't it a beautiful view? Sun's kind of messing it up, but I was here a while. Until next time.